Good evening. I'd like to start by thanking the organizers of Congreso Futuro for doing a wonderful job of this conference again, and for inviting me back. It's always a pleasure to be here in Santiago. I also realize I'm the last thing standing between you and dinner, so I will try to keep this brief, but also this talk is about the relationship between our food, our microbes, and our health. So perhaps you will think of some new things about dinner as you go to eat after this talk. So what I'm going to talk about is some of the key results from the microbiome overall, especially in the 5 years since I was last in Chile, and also some of our more recent research on healthy aging and AI. When we think of what we are, often we think we are what we eat. And this idea is not new. So, for example, the Italian artist Arcimboldo painted this painting back in 1591. And then, in 1863, the German philosopher Feuerbach expressed the idea as der Mensch ist, was isst, or the person is what he eats, which I guess is a much better pun in German than it is in English or in Spanish. But we have this intuition that what we eat affects both who we are and also how we are. Now, back in 2016, I had the pleasure of meeting Jeff Bland, founder of the Personalized Medicine Lifestyle Institute and one of the fathers of functional medicine. And I was struck by this quote from his talk where he said that food is a language that speaks to our genes. So think about what that means. Our human genomes are fixed at conception, but what our genes do, how they're expressed, depends on what we eat, whether we're going to be thin or fat, strong or weak, whether to give us a long and healthy lifespan or to consign us to a short life full of disease. And it's increasingly recognized that this language of food is the language of color. I'm sure you have heard the idea that you should eat the rainbow, and that idea is a very good idea. And what's going on behind the scenes is that this language of color is expressed in chemistry. So everything from the carotenoids that make the carrots orange to the anthocyanins that make the berries blue are specific plant natural products, and we're just starting to understand how they affect how our genes and how our microbial genes are expressed for health. Now, eating right can be difficult, and even where I live in California in the United States, the closest source of food to my house is regrettably this one. And I showed this slide 5 years ago, and it hasn't changed. The sad thing is their motto, which is too much good stuff. So when I take my child there, she is surrounded by bright colors, by strong flavors, but you might think they stripped out all the good stuff and replaced it with synthetic analogs with completely different impacts on your health. And unlike Chile, where Senator Garrity introduced this wonderful labeling law, there is no law like that in the United States to tell you which of these foods are really bad. But we're learning it anyway, and so articles like this one are coming out in the American Medical Association, saying things that you do not want doctors to say about something that makes up 60% of the American diet. Like, the health consequences are dire, and the concerns include type 2 diabetes, cardiovascular disease, and dementia, which I'll say more about later in this talk. However, if you've been following the popular press, you know that there is a simple solution to all of this. And so at about the same time, this article was published saying that cheese protects you from all causes of death, says science. And that's great, right? Because why would science say something like that if it weren't true? Finally, humanity's battle with the Grim Reaper is at an end, right? All we had to do was fight him off with the right piece of cheese, and we'd be good. Now, sadly, it's not quite so simple, and nutritional science is really hard, and here I'm highlighting what's still one of the largest studies of this type, looking at weight loss and weight gain in 120,000 people over 20 years to find the relationship between food and health. And I have to say the results are not surprising. So when we think about the foods that we stereotypically think thin and fat people eat, the food that caused most weight loss was yogurt, no surprise there, right? And the food that caused most weight gain was chips, again, not very surprising. But what did surprise me was how large the effects were, or rather how small. So each serving of yogurt per day led to weight loss of 372 grams over a 4-year period on average. Whereas each serving of chips per day, they gained weight of 768 grams. So think what this means. If you give up the worst food every day and you eat the best food, and you do that, make that right choice 1,460 times in a row, it's only going to change your weight by about 1 kilo. So you might say, well, that's not worth it. I'm just going to eat the chips and gain that kilo. But what this is masking is individual difference. So some people make that change and they lose 20 kilos, whereas others make the change and they gain 19. And almost all averages out at the population level. So we need to understand individual variation. Now, I've been talking about weight, but our food also affects our mood in ways that we're just starting to recognize. So in the SMILES trial in Australia, they wondered, if you have major depression, could you cure it and be happy with diet? And so they tried Mediterranean diet, and they got a 32% recovery rate, which is comparable to SSRIs like Prozac. Now, to check it was diet-specific, when they used low-fat diet as a control, only 8% of them got better. So this is pretty remarkable, and in a later study, they showed it was as good as group therapy, current non-pharmaceutical standard of care. But even if you can get 32% of people to recover, that still means you have a 68% failure rate, and it doesn't work for most people. So we need to understand why. Now, there's been a huge industry looking in the human genome for the key to diet and health over the last 25 years. And billions of dollars have been invested in nutrigenomics. But despite all this, there is no test that you can do today that will sequence your genome and tell you what you should eat at this kind of level of detail that you need to make good choices. So I start to wonder, are we looking in the right place? Or are we like the proverbial drunk man looking for his keys under the streetlight, not because that's where he dropped them, but because that's where the light is? And I start to wonder, are all the keys to this critical health problem somewhere completely different where we haven't looked yet? So to find these keys, back in 2008, my lab introduced a new method for improving DNA sequencing of microbes. And in one experiment overnight, we collected as many microbial marker genes, a kind called 16S ribosomal RNA, as had been collected by the whole scientific community over the last 3 decades. And we looked at microbes from lungs, microbes from rivers, from the air, even from the most complex environmental microbiome that we know of on the entire planet, the Guerrero Negro microbial mat. And what you can see is that they cluster together beautifully. The same kind of sample clusters together on this plot. But I won't go into much detail on this, but what I do want to focus on is the cost, because we collected 500,000 sequences that at the time would have cost us $8 each. So if we did this with old technology, it would have cost $4 million, but we did it for just $12,000, and we did it overnight instead of in a decade. And so this is what led to the Human Microbiome Project, to the Earth Microbiome Project that I co-founded, to the American Gut Project that I will tell you about soon, and many other projects. But what is most amazing is that sequencing that cost $4 million in 2007, that we brought down to $12,000 in 2008. Today, we can do it for just 75 cents. And this radical decline in the cost of sequencing is what lets us find out so much about our microbes. So in the Human Microbiome Project, which I talked about here 6 years ago, we started to make these maps of microbes around the human body. And you can see that different people have not one microbiome, but many different ones, almost like different continents on this map, that we can use to understand links between microbes and health. When you think about DNA, what is amazing is how many microbial genes we have. So each of us has about 20,000 human genes mapped by the Human Genome Project. But amazingly, the Human Microbiome Project and MetaHit and others show that we have from 2 to 20 million microbial genes. And by this measure, you could say that we are just 1% human in terms of the DNA we carry around with us. And we ignore those 99% most of the time. And what is shocking to me is that these 99% we ignore are the ones that we can change. You change them as you had breakfast this morning, you will change them as you have dinner tonight, you change them as you develop from a baby, and if we can take control over that process for health, we could have radically better outcomes. Now, the vast majority of these genes are bacteria in the gut, but it's important to remember the gut is not Vegas, and that what happens in the gut does not stay in the gut. And what we are learning is that our gut bacteria communicate even with the brain to impact behavioral and psychiatric and neurodegenerative conditions. And what we are really excited about is that future therapies for psychiatric disease may treat the gut, not the brain, including with technologies like the phage therapy that you heard about earlier in the session. And so we're learning how the gut microbiome affects processes throughout your body, including things you expect, like digestion, things that are more surprising, like the immune system and clearing pathogens, including even SARS-CoV-2, and then in brain development and behavior, especially when we're very young. But it can also go wrong and have negative effects, including things like metabolic syndrome, diabetes, obesity, which are still becoming more prevalent here in Chile, GI disorders, cancer, and neurological and neurodevelopmental conditions. So you might think the microbiome is so complex in what we can do. And in the American Gut Project, we turned to citizen science to look at many factors that affect the microbiome. And what we found is that a very simple thing, the number of different species of plants that you eat, the diversity of plants, is key to changing the microbiome. And one of my colleagues at UC San Diego, Larry Smarr, took this to an extreme level, making a smoothie out of 60 plant species and drinking it every day for months. And this was featured on the Netflix documentary Hack Your Health recently, if you would like to find out more about it. Now, I'll be honest, right now using this microbiome data is really hard, and the presenters had a terrible time trying to figure out what all these complex displays meant. It is easy to understand lots of food, it is difficult to understand lots of microbes. And this figure is a little technical, but essentially what we found is, unlike most people whose microbiome stays the same, Larry became 5 different people with different microbiomes over 8 years, and the last change was a change with the green smoothies, which are the dots in green, which made his microbiome look like the microbiome of a new person. So you might say, so what? And what we can couple it to is things like his health parameters, like his blood glucose. So he had been gaining weight and transitioning to metabolic syndrome for several years, and becoming a vegan and eating a gluten-free diet did not help him, and that is the last three months of this plot. But immediately, the green smoothie brought him back into the healthy range for his blood glucose, and his visceral fat decreased by 30%, and he also lost weight. And so we are trying to understand whether this kind of simple intervention can have important effects for more people. To build on it and to make this work for more people than just Larry, we are involved in this large-scale project called Nutrition for Precision Health, which NIH is spending about $170 million on in the United States. And the idea is to build AI algorithms to tell at a detailed level what particular people should eat based on their microbiome, their genome, and other parameters. We're also specifically exploring Alzheimer's disease, and like I told you, there is this connection between the gut and the brain, and for the most part, we are piggybacking on clinical trials of diets that we know modify Alzheimer's risk. And there are many successes in this project, but perhaps the most important one is this concept that we can use source tracking of diet in molecules in the blood. So if I run your blood through a mass spectrometer, 95% of the molecules no one knows. But if we run all these foods through the mass spec, we can say, I don't know this molecule, but it comes from an apple or a salmon or some other source. So then we take those food counts and we match them to performance on memory tasks. And I apologize, this is a little technical, but essentially what we're looking at is foods that are bad or good for memory. And if we look at the foods that are worst for memory, what we see is sugarcane and soda. And if we look at the ones that are best for memory, we see fish, strawberries, and mushrooms. But you might say, so what? I could have pulled names of foods from a hat, and it would be just as convincing. But when we look to the scientific literature and the popular press, we found thousands of articles about how soda hurts your brain and causes Alzheimer's. And we found the same for sugar. So if you are thinking about having sugar in your coffee at dinner, perhaps you will skip it. And similarly, at the other end of the spectrum, we see a huge literature on how good fish is for your brain. And we even found some work showing that both strawberries and mushrooms are actually good in the specific context of Alzheimer's. So what is exciting is, just like the advance in sequencing, now we take thousands of person-years of research and turn it into one afternoon on the mass spectrometer. Now, I do want to comment that it is still true that microbiome data is dominated by the United States and by Europe and somewhat China, but you can't even see South America on this plot that was in Science a couple of years ago. And I want to say that the technology of sequencing and the technology of mass spec to read out the metabolites makes this kind of thing accessible at price points that are far cheaper than developing the technology in the first place. And so putting Chile on this map in a serious way would be an amazing goal. But having the data is not enough, and the real challenge is to build a user interface for our microbiomes. And so I showed you the technical complexity of Larry's data, and what we want to do is we want to make it so that you can do it with your smartphone. So the idea is, when you are confronted with 1,000 yogurts in the supermarket, you could scan it and see, is it good for my health or not? And that you could ask that question in natural language. So I'm sure you have all used ChatGPT by now. We're calling this something slightly different, but related. And the idea is that you could ask your phone in natural language, in your language, should I eat this to improve my health? Or can you highlight with augmented reality what I should eat instead of this one? And our goal is to make this work everywhere. So the idea is that even at a market anywhere in the global south, it should be possible to do this. For example, to support pregnancy, which is the best way to combat childhood malnutrition, where you could perhaps ask, should I eat this produce or that produce so my baby will be healthy? And this might sound crazy, but smartphone penetration will be 95% in India by 2040, and Bangladesh is only 10 points behind that. So we think we can develop this technology right now, deploy it in wealthier countries, and then deploy it everywhere, just as cell phone technology has penetrated everywhere to benefit health for everybody. So that is the kind of stuff that we're up to at the Center for Microbiome Innovation that I direct, with an increasing list of companies, an increasing list of faculty members who are part of it, and an increasing number of collaborators around the world. And to build this global system combining microbiology, chemistry, and AI, it is something we really need to solve as a global problem together. And so I certainly look forward to more discussion about that. You can find more about all of this stuff in my books and in my TED Talk. And once again, I would like to thank the organizers of Congreso Futuro for all the hard work that goes into making this conference a success. And I wish you all a good dinner with some thought-provoking ideas about how you can use that opportunity to change your microbiome for good. Thank you.